There's a big group around the computational biology, and then on the right side, we do a lot of drug discovery as well. And I mean, some of you may be familiar with this, but there's a company called Infenza Bioscience that was spun out a couple of years ago. We had a little to try to prevent cells from dying in a heart attack. So that's now where we're moving that forward for clinical testing. And so that's another area where we're doing a bit of drug discovery. So there's a bit of interdisciplinarity here in the research group, and that's there for a reason, because to try to study really complicated questions, you really have to bring people from a lot of different disciplines together to try to solve the problems. And so that's really where we have been focusing around this particular consortium. So let me just start by asking a basic question of all of you, like in regard to your current year level. Let me just start by seeing who's in like year 1 undergraduate. Anybody? Year 2. Okay, a few people in year 2. Year 3. Okay, a few people, and then honors level. Okay, a few honors, and then PhD. Excellent. Okay. Anybody else I haven't covered on those levels? Yeah.
Masters.
Masters, of course. Master's level. Anybody else? Okay, so we've got basically, what you can see here is we've got everybody from, looks like second year undergraduate through PhD, anywhere in between that, a broad spectrum of individuals. Does anybody have, like just from a show of hands, familiarity with pluripotent stem cells? As in you've had a lecture about them, or you kind of know what they are at all? A few people. Okay, yeah. And then anything, trying to think of like general cell biology. I think probably everybody must have some general cell biology familiarity with that, right? Okay, so that's kind of like a starting point. In this context, I just want to be clear that our interest in bringing you into this consortium is to, number one, give you all an opportunity to be engaged on a research project that we're trying to accomplish, which we can't possibly do without you. And so for that reason, there's a bit of like sort of partnership here that we're trying to build to try to develop something that, you know, among my lab and the three of us in particular, it would just be simply impossible for us to do this. So this is why we're kind of going to be running an experiment alone, kind of separate. Like we've got the science question over here, but the other question is, can we get a whole bunch of undergraduates and PhD students and master students to come together, given all of the variation in your training, to help contribute to what we're trying to do over here on the science side? And that's a totally different question. And so I just want to be really transparent with you, which is you're going to be getting communications from us along the way. We're going to try to be training everybody up a little bit so that you've got some knowledge about what we're trying to achieve. For some people, what we're going to do is like just stupid simple. They've already been like way down the road over here. Whereas for other people, that might be really challenging to try to like learn the skills that we're going to try to get you to develop in the course of this. So there's going to be a lot of learning from our side as well as from your side in this context, and we're going to just be experimenting on both getting the consortium to work together and then also trying to accomplish a research project. Now, the reason why this is exciting for me is partly because there's an aspect of, I've had a hope since I started my lab here at IMB 10 years ago. I thought, surely there's a project that's going to be able to get my word out to a bunch of people, and we're going to sort of leverage the sort of minds and capabilities of a lot of people around who are interested in doing research, doing something that would not be possible in my own research lab. So this is something I've had a hope to do for like a long time, and so really excited to be able to kick things off. Now, let me just sort of start with a big picture, and then I'm going to have Nick and Chris give some of the more detailed aspects around this. So in my field, like we're interested in using stem cells to make to do different things, like to make heart muscle cells or study diseases like ischemic heart disease or arrhythmias or other kinds of things, drug screening for those, or understanding what's going wrong in the heart. And in this context, you know, there's been like 20 years of people trying to do this, but everybody kind of works in their own little space, and there's very little ways of being able to connect the information. So this project is really a hope that we'll be able to consolidate information that has been sort of out there for 2 decades but never brought into one place. In order for us to mine that data so that we can make better protocols that allow us to differentiate cells into the heart. Now, the even more grandiose hope from this is that we'll be able to do this for literally every tissue and cell type in the human body, okay? So that's where we're going to be starting with the heart, because that's what we know and that's what we're familiar with. But this is where I'm hoping you guys will, if we can get things kind of up and running in the, things well-oiled machine kind of situation, the hope is that we'll be able to jump from like a ventricular myocyte and then into an atrial myocyte. We're still in the heart and still cardiac physiology, but what about neurodegenerative diseases and that whole lineage? So big challenges of trying to differentiate into neurocells or into the liver or into the pancreas. So there's a lot of knowledge out there, and it's very stochastic and spread out. And so we're trying to use this consortium to create something that would be an incredible resource to the globe in regard to the ability to work with stem cells more effectively. So the way we're going to run this today is Nick is going to give you a little bit of the background around iPS cells and stem cell differentiation and what that's all about for those of you who may not be familiar with that space and what we're trying to do. And then Chris is going to give a little bit of his background on the data sciences and development of this data mining resource capability. So that's going to be a little bit here for you guys to get up to speed, and then we'll just have a bit of a discussion about what's going to happen. All right, over to you, Nick.
Okay, thank you. First of all, I just give you a very brief introduction my career so far. I've completed my PhD in cell biology at David Ferris lab at IMD. Some people you may know that. So at that time, my research was focused on metabolism and immunology. So basically, I used small molecule and biochemistry techniques and a couple of cell and animal disease models like inflammatory bowel disease, obesity, diabetes, and cancer to identify some GPCR drug targets and to study the mechanistic signaling pathways. After my graduation, I moved to Nathan's lab. At this moment, I'm interested in using stem cells to optimize different cardiac disease modeling, like Nathan mentioned, diabetes, ischemic heart disease, dilated cardiomyopathy. Also, I'm developing preclinical models of diabetes and ischemic heart disease as comorbidity studies. The other aspect of my research is to identify some drug targets for treating ischemic heart disease, like different ion channel modulators. So you will see, in 2012, the Nobel Prize for Medicine was awarded to 2 stem cell scientists. So John Guten and Yamanaka, sorry. So they were jointly recognized for discovering that mature cells can be reprogrammed to become pluripotent. This technique is well known as stem cell engineering induced pluripotent stem cells, iPSC. Over the last decade, human iPSC technology has been advanced a lot to different areas like disease modeling, drug discovery, and also for regeneration medicine. So the other important area for stem cell study is to basic research and developmental biology. So for example, we can use human iPSC to study early human development and cell fate decisions. And we can also investigate some gene functions, epigenetics, and the cell signaling pathways. As Nathan mentioned, our lab is focused on heart and cardiovascular disease, because this is the area we are good at and we are famous for this area. So basically, undifferentiated iPSC carriers, they can be differentiated into different lineage, different mesotome or liver mesotome, and then later stage to different progenitor cells and endothelial cells. And so here you can see our lab is using one of the different cardiomyocyte differentiation protocol, which we use small molecules and to induce human iPSC to cardiomyocytes over a 15 days window. So you will see the top right as a beating cardiomyocytes, and the right side you see some immunofluorescence of the cardiomyocytes marker alpha actin and the nuclear. Human iPSC cardiomyocytes in vitro model is perfect for different utilities. So over the last 10 years, since Nathan moved to UQ our lab has published a couple of papers using human iPSC carriers to study, for example, cardiac physiology, like electrophysiology, calcium handling, contractility, metabolism, and also use these human iPSC carriers to model different cardiovascular disease. And also we use these different disease models to study drug development and regeneration. Why we use human iPSC carriers? Because they have different benefits over other non-human iPSC carrier models. So for example, they have more closed myocardial gene expression to human adult cardiomyocytes. They also have well-organized contractile components, which is in terms of the structure of these cells. And these iPSC carriers are more response to physiological and pathophysiological events, like response to ischemia, to response to arrhythmia. We can have multiple preclinical models and clinical regeneration applications in cardiovascular disease. All right. To better reflect how we address the key challenges of human iPSC carrier studies, so our team is working on the database. We developed a well-known human iPSC carrier portal. So now I will pass this message to Crystal. Crystal will give you a detail of this database.
Thanks, Nick. Okay, so first of all, thank you everyone for coming. I want to introduce a bit of my background because, one of the feedback that we got is that some of the students were feeling intimidated, scared that they are not going to be able to participate in this consortium. And I just want to reassure you that me being here talking to you is way more scary than you guys being part of this. So just trust me that this is going to be a really casual, friendly consortium event. And building on the ideas that we have highlighted, I kind of want to highlight the reasons we are doing this consortium. First of all, 1 in 2 females and 2 males develop some form of heart disease in our lives. It affects a lot of us, and we don't have any existing drugs that reverse the permanent damage caused by ischemic injuries. And these cells, the stem cell-based cardiomyocytes, heart muscle cells, they're really there for us to act as a model for us to better understand heart biology, discover new mechanisms, and also model the new drugs' safety and efficacy in hearts. These are ethical models because they are not animal models. This is really important because in the European Union, they've actually started to ban all animal models in the clinical studies. So which means that these models are going to take traction, and it's really important for us to use them. So what are the challenges in using these models? Now, we want to mature the cells because all stem cells, by definition, they are embryonic states, right? And we are trying to mature them by designing different protocols. So we have kind of identified what are the exact protocols we need to use in order to differentiate a stem cell into a cardiomyocyte. But the problem is, we don't really know how these protocol variables are impacting every single aspect of the maturity. Because maturation is not just one single element, like there are many aspects to it. One of the more important ones that we typically think of is the functional properties, like how the cell contracts or what is the contract rate. And then in other aspects, you have the electrophysiological properties, which is really important as well because they are highly related to drug modeling. They use sodium calcium channels, and these are the channels that the current drugs are targeting. And we also have the morphological and structural properties as the other category. So this slide is a massive table, and I really just want to highlight one point here, is that there's a huge difference between immature and mature cardiomyocytes. You can just imagine that by having like a baby heart versus an adult heart, the way they contract or the force they have is completely different, and the mechanisms that underlie them require different drugs to cure them if you have a damage. And there are a lot of ways to mature these cardiomyocytes. The problem is we don't really know how, which one is the best one and what they're really doing. Each of these figures are a single maturity aspect, and as you see, although if you look at the first 3 graphs on the left, you can see that although we are getting closer and closer to real adult values in the human, like in fetal, in our current hearts, we're still quite not there yet. So that's something that's like an ongoing challenge that's really significant for us to solve. And imagine when animal models are gone and you're relying on these cells, like what can you do? So I really didn't come from a biomedical background at all, which is why, looking back in hindsight, I've learned about stem cells 1 year from now. I see some familiar faces. Some of you might already know that I didn't learn biomedical during my undergrad. I was in industrial biotechnology, learning chemical engineering, how to deal with bioreactors, calculations, so I was nowhere near the space. And in the past year, I really started to understand the difficulty in these brain cells and having different aspects. So what I want to show here is that if you think of the Pokemon game, you have these berries which you can change the statistics, like the attributes of Pikachu, right? You know that this berry will lead to high attack and that could lead to high HP. But in the case of cardiomyocytes, we simply we know that these protocol variables are leading to the cells becoming into cardiomyocytes. But what are they actually doing? Like you could have a completely opposing effect or a synergistic effect, where you could have higher contractile force, but a worsened beat rate maturity. So is this cell more mature or less mature? Like we really do not know. And as you see on the right here, I show you one of the figures, which I found really interesting, because protocol 20, these two protocols, one of them is published in European Heart Journal, one of like a crazy science journal, which impact factor of 50, like compared with the Nature papers. The other one is Cell Stem Cell, which also has one of the best journals in the field, but that one is purely for stem cells. So if you hear the name, you kind of understand that one is more a translational journal, while the other one is purely scientific. And the maturity aspects are completely different. So during my honors, to re-summarize this, is that we have decades of cardiac differentiation knowledge since the Nobel Prize winner, right? And we have hundreds and thousands of people working on these cells, trying to differentiate cardiomyocytes, and having many, many studies all together. And there's one systematic review that's made those figures that we showed earlier, and they compiled all these studies into one single Excel spreadsheet. So these people, like these researchers, are from Harvard and MIT, and their conclusion is that these are one of the smartest people in the world, and their conclusion is that it's really difficult to find the actual impact of protocol variables. So which is why we built a machine learning analysis on this single data set, to try and understand this, to really understand its impact. And I've built a website during my honors, which allows you to dynamically navigate the protocols. And it's currently publicly available, so you can also try and have a play. One of the challenges is that I kind of now understand that it's a bit hard to use and it's not that intuitive. So if you want to learn more, that's something you could discuss with us definitely and make suggestions and work on this together. To give you an idea of the challenge involved in this is that this will be the original Excel spreadsheet. So I'm just skip all details and show you what it looks after. And as you see, there's a huge difference in terms of whether this data is good enough for machine learning. So that is something that we're going to try and achieve together. We're going to make one of the world's first student-curated. And this has never been done before. Having a student-led consortium project which aims to create any sorts of data that can be used scientifically at a high impact level. This is going to be the first. And part of the reason I think we are in the best time now is that we have ChatGPT. We have AI to assist us, and we're going to try and use that in this consortium. So this is an example of the berry-like thing in Pokemon, where you have different protocol variables on the y-axis, and then you kind of get a value of their association with the outcomes that you're trying to achieve. And the takeaway of this graph is that, on the x-axis you similarly have the protocol variables, and the graph on the top shows what protocol variables are associated with contraction strength. And on the second plot downwards is what studies are being used on ischemia, which is one of the leading heart diseases. And you can see there is a clear knowledge gap in the metabolic part, like the orange part, where there's pretty much no enrichment at all down there. And therefore, you can use these kind of methods, like it's a real case application that we are also trying to make a paper out of, to just kind of understand where it is going and what is the impact of this. So on that note, I'm going to pass this back to Nathan. Thanks.
Okay, so this is where we're going to just give a bit of a sense around what you guys are going to be involved in on the side of helping us curate this information. So the bottom line is that we've got, as you can see from the portal, and you guys can have a look at it, you can read the paper on bioRxiv if you're wanting to get a bit of a sense about what we've done so far on this. We've only focused on ventricular myocytes, which is where we've got most of the data available to us. But now we're wanting to start to expand that and start to move out into new cell types. The heart is going to be our initial goal, is just to build a cardiovascular atlas of information, and then we'll just keep on going depending on how well things are traveling with everybody on this one. So the goal here is, your participation is going to be around fundamentally building the database. And so by default, that means that you're going to be reading papers, and there could be a variety of ways. As Chris was mentioning, you could use, you might use AI tools if you're not familiar with certain things, if you don't know about them, and you can use a variety of methods. Ultimately, we've got to go from a bunch of students working on curating information with different levels of knowledge into a data resource that is high quality. So we've got to do this QC check to make sure that people, as you're learning and diving into reading and curating the information, that we are not necessarily just throwing it into the database because it's there. It's we'll have QC checks, there's going to be training that's going to go on, and there's probably, because people are PhD trained in this and maybe masters, and even at the undergraduate, you might already be familiar enough to be able to just jump right in and read papers and curate the information. For example, if anybody in my lab were doing this, I wouldn't need to train them on anything because they could just simply fill out the database. But for others who are not familiar with this, there's going to be more training that's going to be involved because there's a lot of it's like you're reading a whole new language. What is a Wnt inhibitor? What is FGF? What is Matrigel, right? You're going to have to learn about what things are in order to know how to add it into the database. So this is where we'll have a bit of work around trying to make sure that we're accommodating that broad range of expertise within everybody's training expertise. So the goal again is to get this big database, and we're going to be training you guys around protocols for differentiation, how to read them. And then ultimately, as I've mentioned before, the goal is to, have the consortium publication that would be allowing people to have authorship on that paper as we start to get that paper out. And you can now use that as your own CV builder in regard to having experience in a research project. And I want to add on to this that, from my vantage point, there are multiple ways you guys could get involved in this. As Chris pointed out, this is a lot of data. There is a ton of data that we have available to us. So if you're interested in exploring entry sort of aspects to this, is helping us curate the information, because that's ultimately what we can use to try to develop and study that. But if you want to actually work with us to study it, maybe developing, if you're interested in computational biology and you want to work with kind of data analysis methods, this is where your involvement could come in at multiple levels. And I will say that I'm going to be working with some of the course coordinators to help incorporate this, possibly even in terms of credit for coursework, because I think that's another area where you guys can potentially benefit. So there's a lot of exciting directions that we're going to be taking with this going forward, and so you guys are a little bit of the guinea pigs on this one, but I hope you're just as much interested in helping us get down the road with this one. Now, the other aspect you guys have heard possibly is that we've got Stem Cell Technologies on board with this, an industry partner. It's one of the world's largest companies that's focused on stem cell reagents, and we have regular meetings with them around the portal and around the consortium in terms of our data resources. And they're really interested in engaging with us, because the more we can learn about stem cell differentiation, the more from their company side of things there's a benefit from them learning how to optimize protocols. So that's another angle, is actually getting industry partnerships incorporated into this so that we get good uptake for people around the world that are going to benefit from this. So I think those are just the key points. I did want to leave plenty of time for questions. So I think we have, how much time do we have? Half an hour, 20 minutes or something like that. So why don't we just open it up? I'm sure there's more pieces if people want it as a starting point, but maybe let's just see what people's questions are, and we can go from there. But you look like you have a question.
Yeah. How will this data be delivered to us?
Yes, okay, how will the data be delivered? Okay, so that's a good question. What's our tentative game plan here? How are we going to start going into this? All right, so at the moment, the idea is that I think we're going to send a bit of a survey around, because I'm noting that there's a lot of people from different levels of expertise. Like if people are PhD trained versus second year undergraduate, there's differences, and we need to be engaging with you a bit more in a way that doesn't necessarily group you all in one space. So for example, PhD trained people may be just ready to jump in, read a paper, familiar enough with this that we can quickly get them engaged in rolling out data uploads. For those that need more training, we might start very basic, like what is a differentiation protocol? How, like literally sending out some Zoom videos that kind of give you a little bit of a crash course on iPS cells, crash course on differentiation, a crash course on what are the components that are, how do you read a paper in order to curate that information. Now, let's just say from a training side, there's going to be a few things in that space, and we might have you just self-report to us where you think you're at on this, in order for us to kind of begin to tailor our communications. Now, the bottom line is, like, we've got this training component, and then we've got the database. So as I mentioned, the database has got to be it's got to be good, top quality. So the way we're going to do this is that Chris has got, we're working on developing a way in which we'll have, we've already got 300 studies from our initial database. So what we're going to do is we're going to send you, say, 3 papers, and we're going to ask you to curate the database components for those 3 papers. And then we're going to QC your answers against our existing database to see if you're accurate. And so there's a quick way in which we'll be able to quickly assess how you're going in terms of that. If you are good to go on that front, then we can move you into the data curation space. Now, that being said, in the data curation space, that doesn't mean that everybody's perfect on curating new protocols, because people might make mistakes and you might have differences of opinion about what is being reported in a paper. So even in the data curation component, if people are through the QC checks and you're in the data curation component, what we're going to be doing is sending the same paper to multiple people. In the maybe 2 to 3 people will have the same paper. So let's say among, I don't know, say 100 students that are going to be curating information, 3 people read the same paper and they're all in great alignment, perfect. Then great, let's just check that off, it's good to go. But maybe all 3 of them really don't have the same agreement. And that may not be your fault. It may be that it's a crappy paper and they didn't write the methods very well or report the information. Could be a variety of things. But it'll flag for us that this particular data isn't really good, or we need to double check it before we actually accept it. So we've got multiple steps where we're going to be training and QCing people's ability to then incorporate and curate information. And even in the curation process, we're QCing that whole process to make sure that all of the data that's finally received into the full portal is got multiple levels of QC checks. Does that make? It kind of runs through the whole pipeline, I think, at least as we see it at the moment.
Ethan, why don't you touch on a bit on the papers that we are aiming for?
Okay,
What
yeah.
kind of studies we're trying to do about this?
Right. So the papers, we've got 2 papers in mind for this. The one paper, it covers both sides of this. One paper is going to be around the development of a community-oriented data curation sort of mechanism. So that's basically the consortium process of how we're engaging with you, how we do the QC checks of the data, and we're going to be curating data around that as you sort of feed it to us, so we can build a paper around how to develop consortium scale data curation. And so that's one aspect. And then the other one, obviously, is going to be around the data and what we're going to be learning about from that. So the one thing I'm so excited about this in regard to what we've already found with the current database is that there's this notion, just to give you a little bit of a sense about this, in the field of stem cell differentiation, that you have an adult cardiomyocyte over here as being like the grandiose vision. Everybody wants to make an adult cardiomyocyte. And so everybody's out there finding a way to say, my protocol is the best one for making that cell type, right? Now what the portal, everybody's got a different opinion. They'll add FGF here, they'll add ZAV here, or IWP2 here, or add insulin this way, or whatever the case is. They're going to be throwing the kitchen sink at the cells and saying that theirs is the best one. But there's no way to benchmark your protocol against everybody else's protocol. And that's a big problem, because they just make a claim that theirs is the best thing. But there's no way to say yes or no. So the great thing about the portal is now we can properly put head-to-head any 2 protocols and see how they perform in the context of these types of things, like how well does a protocol make contractile cells compared to any of the other protocols. And here's our key take-home point, which we have now begun to really validate out, is that no protocol is the gold standard. There's no protocol that can make the best adult mature cardiomyocyte. Every protocol is good, but they are good for different reasons. Maybe a protocol is good for studying ischemic heart disease, but that protocol is not going to be good for studying like Brugada syndrome or hypertrophic cardiomyopathy. But we can figure that out. Now that we've got the data, we can actually say, if you want to study ischemic heart disease, you're going to be wanting to look in this territory. These protocols are the ones that are really great. And these ones are good for hypertrophic cardiomyopathy or Brugada syndrome. And we can guide people more directly toward the protocols that are suited for the purposes that they're trying to actually accomplish, rather than simply saying that there's one protocol everybody's trying to get to. And the other thing that's surprising about this is that the things that drive contraction are not the same things that drive the electrophysiology of the myocytes. So for this reason, they don't fall into line with each other, the mechanisms that drive maturation. And that's why you've got this sort of broad spectrum where you need to have a specific protocol that you're going to be using for disease modeling. So that's why this portal is super helpful, because we need to be able to do this for every cell type, because I have no doubt that it's generalizable. But there's no way to do it unless we curate the information. Other questions?
How does the database compensate for differently derived iPSCs? One from just healthy and different ages,
Sure.
but also for disease modeling.
Yeah, so the difference is in the iPSCs used for the protocol and the differentiation, basically,
Yeah, so
or account for the method of reprogramming.
one for like a healthy control patient, but two if they're derived from someone, so say you're looking for someone who's predisposed towards ischemia, looking at the different iPSC origins and seeing which ones are more prone for different disease modeling.
So on that point, I've got two answers to you for this. The portal currently, when it comes to curating data on differentiation protocols, at the moment, we're only curating information on the control cells, not on disease or mutation. Maybe that'll change down the road, but for the sake of getting generalizable information, we're only using the information reported for the controls. And that being said, for example, if a study is reporting a protocol used for ischemic heart disease modeling, for example, we don't report on the ischemic heart disease variant iPS cells, we just report on the controls, but we indicate that it's being used for that purpose, and that helps us then link that information in the portal. Now, that being said, the other thing is that I think is related to this is like, what if people use, across the world, people are going to be using cell lines, hundreds of them. Like, nobody's using the same cell line, right? Sure, they are using the same cell line, and there's a lot of them that are used commonly, but there's still a huge diversity. So how much does a protocol be affected by the cell line? It's not a protocol issue, it's a cell line thing, right? That's a big, big challenge. Now, the beauty of this is that oftentimes protocols are going to be used across different cell lines. So the same protocol is going to be sampled in terms of its underlying effects on multiple cell lines that might be reported in the database. And I will say, as an example of this, one of the things that I was surprised by, but it comes out in our current analysis, there's a cell line called BJ1 that is apparently very, very strongly positively associated with really contractile cardiomyocytes. And I don't know, I've never worked with the BJ1 cell, but the point is that the database can actually look across all of the information and show you that even a cell line is actually a reagent. It's a component of the protocol, and it will report on whether a specific cell line is more useful for some purpose than another cell line, if it is in fact enriched. But those are great questions, yeah. Other questions?
Or any question from Zoom? Not yet.
Yeah,
What's the expected timeline of this project?
great question. Another good one. Expected timelines and activities, and sort of like how things are going to go. All right, so the answer is it's going to depend, because like maybe for people that are going to be ready to go immediately, we'll be ready to jump straight into it. For others, it might take a bit of time for training. So like I can't give you an immediate answer, but this is also, it's obviously a volunteer thing. So like your priority is your coursework and or other things that are going to be going on. And this activity, ideally it kind of sits in time where you've got availability to kind of work on it. And that's why we need to have reasonable expectations about not overloading you, especially if there's exam times or whatever the case is. So in my, like, again, back of the envelope kind of situation, let's just say one of you has gone through the QCs and looks like you're ready to go, and we're ready to curate data. I'm thinking of sending papers out, maybe 3 to 4 papers, maybe, again, a few weeks to read them and report them back. Hopefully you're just kind of like, spend a couple hours here and there where you might be able to read the paper, and then you don't even need to like do it all at once. You can just do it at your own leisure in some regards. My view is that because we've got a lot of students involved, even if, let's just say, even if we've got, say, 50 students and that are into the curation stage, and you're, reading 2 papers, let's say conservatively every couple of weeks or something, one a week or one every couple of weeks, you can imagine across all of you, that's just immediately, we've got a couple of months, we've got hundreds of studies that are in the database. And our current one is 320 something papers.
Yes, 322.
So you can see that it's just a power and numbers thing here, but not overly onerous on any individual. That's kind of the goal here. So hopefully it's all a bit manageable and we'll get you all involved and try to keep it a little bit of a side thing. But again, like I said, you can be involved as much as you want. And you know, I will say, and for those that don't know this, in academic research, there is an element to which contribution is reflected in authorship. And so those are things where we have this sort of baseline expectation about the consortium, and that'll sit within that kind of authorship space. But people who want to jump and elevate their contributions through either contributing a lot of studies and or intellectual contribution to the design of the consortium or anything else that's above and beyond sort of basic expectations, those things are going to be again reflected in terms of authorship positions and other reflections of the contribution to the study. So we're super flexible, interested in having people engage at any level. That being said, minimum is that for authorship in any publication, your contribution has to be seen as being required in order for that to occur. And so by default, that means that you've contributed something that's been incorporated into the database that then is used to mine the data. And so the goal again is that people are going to be at different levels, and we're hoping that we're going to get everybody over to the data curation side ultimately, so that that is reflected in terms of the scientific standards for authorship.
Can I talk about another idea?
Yeah.
Yeah. So as you know, human stem cells can be differentiated. Human stem cells can be differentiated into multiple cell types, like cardiomyocytes, neuron cells, immune cells, liver cells. At this moment, we are focusing on heart cells. So at a later stage, in the longer term, if you would like to contribute more, apparently we have plenty of opportunities to continue this project.
And I think on this point, I did want to bring up, who all has iPS cell experience and familiarity with iPS cells? Raise them high. Okay, so 1, 2, 3, 4. Okay, 5, at least here. Here's a suggestion, I'm just going to put it out there. If you have expertise on non-cardiac lineages, and you would be interested in helping us move the protocols into that lineage, there's a lot that goes into being able to build a database by having know-how around specific protocols. And so we would be very interested in discussing ways in which your domain expertise on differentiation could be better leveraged so that we're more easily able to move the consortium into some of these other lineages where we don't have any expertise at all, but we need that domain expertise. So that would be a really great way just to think about ways in which you might, if depending on capacity and interest, ways in which you could contribute. Other questions?
So Michelle, I think that also answers your question pretty much. And just to give you some of, like if I'm more specific about the actual task, we're pondering a few ideas, and we please understand that this is going to be like a troubleshooting phase where we really don't know what works best because no one in the world has ever done it. So Nathan mentioned 2 studies. The first one is about, the last one is about, that will be like maybe like a year or 2 years down the line, where we're going to have a database. We're going to apply machine learning on it, and we're going to validate the findings. So that will take time. The other study, which is more immediate, is how we're going to establish a student-led cohort to create scientific data. So that is a really, I think the timeline for that is really ambitious, but should be a reasonable timeline would be next June or next July. So it will take like 1 year. So if you are a current student or undergrad, then this could be an opportunity for you to get on paper. And mentioning on like going to the paper point, if you have research experience, I'm sure you know that how hard it is, like how tedious a process can be in terms of getting papers. I just want to touch base on this, just to say that Nathan is not being crazy. because from my own experience, like I started my research journey roughly 2 years ago with him, and I currently have one first author preprint, which is the CM portal study. I have one paper that was published, which is when I first joined this lab. There's 2 more that is currently under press or in review, so really close to submission. And then there's 3 that is close to submission, and 3 that we're currently preparing, and we're getting close to submission as well. So in total, I kind of have like 8-9 papers already in the line. And I just graduated honors last December, and I'm 5-6 months into PhD. So I just want to point that out, because first of all, academics are really egoistic, not Nathan. But the second part is that this is actually something that's feasible, and we can try and achieve this together. The other aspect to this, in terms of opportunities, like if I were you, and I would ask the question, why should I join this consortium after today's introduction session? There are a few reasons. First of all, there's a paper opportunity. The second part is that you gain exposure to how, I guess a bit of exposure to what is required for machine learning, because it's currently the hype out nowadays. What is defined as good quality data? That's really important. And then the other aspect to this is that it's good because you get to know Nathan, be around with academics and see if there are connections that will get you somewhere. The fourth part is that there's an industry partner in this collaboration. And the fifth one is something that I would be personally really excited about, because I was really into entrepreneurship, and I was deciding between whether I should go out, join a startup, or actually become a PhD and study here. And that element is kind of reflected here, because this is going to be the first student-led consortium dataset that's built. And this means that there is potential for us to try and build something that's commercial. And there are companies out there like Scale AI or Outlier.ai. These are companies out there that are creating data for profit. So maybe there's an opportunity here where one way you can provide, you guys are going to be the first people who ever do this, which means that you guys are going to be the mentors, and you can actually participate in this potential business where you can train new people or educate people on how to do this, if that makes sense. So yeah, these are the reasons I would consider if I were to decide to stay in the consortium. And
I have another point on that, which is you guys are.
looking on time.
Yeah. We're hoping that this is really, you guys are the entry point, and this could broaden out quite considerably in regard to the number of students. We're starting at UQ, but you can imagine there's potentially a lot of others within Australia where universities may see opportunities to build and incorporate into these activities. So I think that's where I think there's exciting opportunities, and it's going to be a bit of a steep learning curve for you and for us on this one, but hopefully it's going to be really exciting, and we're going to just keep sending you updates and working with you to try to build and develop some of the capabilities around this as we go down the road. So we'll see how we go. We're coming to the top of the hour. Anybody else have any questions? Yeah.
Will the database be focusing only on IPSCs or including ESCs as well?
Good question. I think we're agnostic to ESCs or IPSCs on this one. I think that's probably a protocol component, is ESL or IPSL. I don't know if it is actually, is it?
I mean, we can add it to the database.
Yeah,
and also, if you're interested in ESG, you can definitely do that. Again, this is a real collaborative thing. We're not going to dictate everything.
Yeah. Good. All right. Well, we're going to go with that for right now. Thanks for hanging out with us for an hour, and hopefully you took some food. We're going to be in touch. We're going to be sending you some updates on emails, and then we'll hopefully start building up as we go into the end of the year and then early next year, and we'll see how things travel. But thanks for everybody's excitement, enthusiasm, interest,